Hello everyone, my name is Kimberlie and I'm going to talk about the effects of algal enrichment and salinity on sediment, particle reworking activity, and associated nutrient generation mediated by intertidal polychaete Hediste diversicolor. And for our core concept of bioturbation, it is biological activities drive ecosystem process, and with particle redistribution and viral ventilation and bioirrigation, and it regulates nutrient cycling and connects bathing pelagic systems. And for our current status, what we use traditional model widely used as a proxy for environmental change. And the big problem we use before our experiment, the traditional models assume species functions are fixed and constant. And for reality, the animal behaviors change with immediate biotic and abiotic conditions. And the relationships between bioturbation and nutrient flux are implied, not directly tested. And it leads to our key question: does a change in environmental content break the link between traits and ecosystem functions? And why we use the model species in our experiment is Hediste diversicolor, and we will call it a worm. And the experiment stresses we use salinity changes times algal enrichment, and our target is to evaluate the activity of bioturbation mediated nutrient cycling under environment stress. And let's talk about the part of methods. We choose the environment site in Eastern Estuarian marketplace in Scotland, and our target components is superficial sediment and the worm and the green alga. Let's talk about the system control and incubation of our experiment. Our core objective is evaluate the individual and interact effects of salinity, algal enrichment, and the worm presence on particle reworking and nutrient flux. And we use 6-day duration. And we use 1.6 liter sediment for aquarium, and our chamber maintained with 12 degrees with 12 hours light and 12 hours dark for cycle. And while we use water management, initial 24 water is spawned off and replaced with board salinity levels for 15, 20, 25, and 30. And for the algal enrichment, we use 4 dry weight treatment levels 1, 0, 1, 2, 3 in aquarium. And for the system stability, we use continuous aeration implanted and no anosmic layers developed at the sediment water interface. And we put 10 individuals worms in one aquarium and statistically encode across treatments. And we use for factorial matrix held in 160 aquaria randomized across 3 rooms. For what we use in imaging systems, we use fluorescent sediment profile imaging, that is FSPI, and we use convert J0 and J5 JPGs to RGB stack to flatten sediment interface. And what we use the mathematical descriptions, like the maximum is the maximum penetration depth to measure long-term downward transport, and long mean and long med is mean and medium depths to indicate of typical short-term mixing, and long rock is boundary rugosity to quantities superficial infernal activity. And what we use to do the experiment nutrient analysis or tagging metrics is calculate net concentration flux over the 5-day experiment period. And we use 20 milliliter water samples collected via 0.45 salinity membrane filters. And that tells about result that the y-axis means mean mixing depth. And we can see that the salinity, in fact, which we can see that is not in fact. And the algorithm, in fact, is significant and non-linear. And we can see when we have 1 gram in the experiment, it is peak activity. That is, sediment mixing reaches its maximum at low enrichment. And when the suppressed activity is at the 3 grams, we add 3 grams. And that's it about the figure 1b. We can see that this is in the superficial activity. And we can see salinity variations cause zero change in surface particle in particle redistribution. And for the algal enrichment effect, it is highly significant. And we can see that figure 1a and figure 1b add 1 grams when it add 1 grams is active for edging, just moving from no algal to 1H significant stimulates surface crawling and mixing. And let's talk about the ammonium interaction. Just look at 2A and figure 2A and 2B. And we can see 2A that it shows a slight home shake trend with ammonia peaking at salinity 20. And we can see the algae effect with low warmth. Adding organic algae has minimal effect on ammonia. And the figure 2b is also like this. Comparing the vertical scales, the ammonia concentrations in figure 2b are drastically higher than those in figure 2a. And that's about nitrate and salinity in figure 2c and 2b. Nitrate reaches its absolute highest point only when there is 0 algae. And in 2D, we can see even at the 0 to algae baseline, the presence of warmth reduces the peak. And in both figures, nitrate is dominant, and wiped out by algal presence, it displays a completely opposite trend compared to ammonium. And let's talk about the phosphate with figure 3A and 3B. And we can see that at salinity 30, phosphate concentrations increase linear as algal enrichment decrease, and so as the figure 3B. Looking at the trends and vertical scales, figure 3A, which is warm absent, and figure 3B, which is warm percent, look nearly identical. And for the silicate part is figure 4A and 4B. We can see with low warm silicates release show a weak response, but reaches its higher baseline on salinity 30. And for 4B, we can see the highest high algal enrichment signature suppresses the driven flux. And looking at the vertical axis, the silicate concentrations is figure 4B are consistently much higher than those in figure 4A. And for the insights for table 1, environmental factors and animal present significantly alter both bioabsorption and nutrient concentrations, but their effects are highly context dependent and do not always interact. And for discussion, activity increase at low levels 1 gram to search for food, but decrease at high levels with 3 grains that we can see at the figure 1A and 1B. And the key insight is the relationship between food supply and biodistribution is non-linear. And let's talk about the figure 2 and figure 3. And we can see that salinity affects the metabolic rate and excretion of the worm. And we can see physical reworking and chemical gradients interact to determine nutrient exchanges. And for the nutrient with silicate and phosphate, we can see the worm enhance silicate released by increasing the reactive surface area through burrowing, and for the phosphate controlled by abiotic desorption and redox conditions relevant for new activity. And my conclusion is the impact on ecosystem engineers is element specific. And thank you very much.